So here you can see nucleotides. Nucleoside, it's two types, nucleosides and nucleotides. You have to remember this because it's basic and it's very important. You have mnemonics over here. See, nucleoside is basically base plus deoxyribose, which is sugar. So side, sugar. Remember? Yeah. So nucleotide is base plus deoxyribose plus phosphate. So this phosphate, so tide, phosphate, so D. If you remember side, your sugar, you will remember the nucleotide also. So this phosphate, which is attached to by a phosphodiester bond, is by 3' to 5'. So this pi dash N of incoming nucleotide bears the triphosphate energy source for the bond. Triphosphate bond is target of 3' hydroxyl attack. So when there is breaking of these bonds, 3' is the first one to get de-attached or attacked, as it says. So the phosphates are two types, purine and pyrimidines. Purine are two rings, as it says, A and G, arginine and guanine. And pyrimidines are three types, CUT, that is thymine, thymine, cysteine, and uracil. Okay. Sorry, I forgot how to pronounce the uracil, so I got confused. Here you have a mnemonic that is pure as gold. So purine, AS, so arginine and guanine, and cut the pi. So PIE, so it's short PY, so it's cysteine, uracil, and thymine. Cut the pi. PY is pyrimidine. So pyrimidines are one ring, and the short name has two rings, and the longer name has one ring. Yeah, one way to remember it. So deamination reaction is how you change. Means when it's undergoing process of the phosphate bond targeting and the translation, cytosine becomes uracil and adenine becomes hypoxanthine, and guanine is xanthine, and 5-methyl cytosine is thymine. So thymine has a methyl group, and CG1 is 3H bonds, so stronger than AT bond because AT has 2 bonds, but CG has 3 bonds, 3 hydrogen bonds. See, so the higher the bond, the more is the melting point, and stronger it's more stronger, and it takes longer to break. So temperature of DNA, higher melting temperature of DNA, and CG bonds are like crazy glue. It's one way to tell you that it's very strong bond because it has like 3 hydrogen bonds. Uracil is only found in RNA, so that's the unique part of uracil, and thymine in DNA. Methylation of uracil makes thymine. So yeah, now you know. Amino acid necessary for purine synthesis. There's one way to remember, it's cat's purr until they gag. So purine has glycine, aspartate, and glutamine. Next one. So de novo pyrimidine and purine synthesis. This is the chain reaction, and it's a various immune suppressive. You'll see it in various immune suppressive, anti-neoplastic, and antibiotic drugs function by interfering with nucleotide synthesis. As I said earlier, that it's a very important thing, because even in pharmacology and oncology, you have to recall all this. uh, so pyrimidine synthesis is leaf flunomide, which is inhibits dihydrotate dehydrogenase, and 5-fluorouracil is 5-FU, if short form, and it's a prodrug. It's a capecitabine form F5FDUMP, which inhibits thymidylate synthase, that is decrease the TMP. Purine synthesis, which is 6 mercaptopurine 6-MP, and its prodrug azithromycin inhibits de novo purine synthesis. Mycophenolate and ribavirin inhibit inosine monophosphate dehydrogenase. Urine and pyrimidine synthesis. Hydroxyurea inhibits ribonucleotide reductase, and methotrexate and trimethoprim TMP, and primethamine inhibit dihydrofolate reductase, decrease in deoxythymidine monophosphate in humans, bacteria, and protozoa respectively. So these two drugs, this one methotrexate and TMP is used in as an antibacterial medications. This is in regards to this one. So CPS1 is mitochondria urea cycle, and CPS2 is cytosol, so used for cytosol, and this CPS1 is for mitochondria. So this one is purine salvage deficiency. So when there is congenital deficiency of purine production or purine synthesis, this is a flow chart where you can see the nucleic acid GMP goes to guanosine, which is a nucleoside, and then it becomes free base guanine. So this happens with regards to HGPRD, with the PRPP. This also causes this PRPP also helps IMP to become inosine, which will later become hypoxanthine. This will become xanthine, which requires XO. Here, these are the medication which stop it from becoming xanthine and uric acid. This is in case of high uric acid in the urine and gout. You use these medication to ilopropanol to stop the formation of uric increase, like deposition of uric acid. And here it's one more, this AMP by APRD to adenine. You can go through this flowchart much better on other platforms which explain you this flowchart very nicely. So the next one is adenosine deaminase deficiency, where you will have ADA is required for degradation of adenosine and deoxyadenosine. Basically, as it says, the word deficiency, you do not have the enzyme required for the degradation of adenosine, And here you have decrease in ADA that will cause increase in DATP and hence a decrease in ribonucleotide reductase activity and lymphotoxicity. So one of the major cause of autosomal recessive severe congenital immune deficiency, SCID. Lesch-Nyhan syndrome is a defective purine salvage due to absent HGPRT, which is hyperuricemia. Gout pissed off means they are usually aggression and self-manipulation and retardation, intellectual disability, and dystonia, which convert hypoxanthine to IMP and guanine to GMP, result in excessive uric acid production and de novo purine synthesis, X-linked recessive. Findings are intellectual disability, self-manipulation. I think I'm not sure, but I think I remember self-manipulation means they eat their own nails. If I'm wrong, just let me know in the comments. I'm here to just read, but if I recall some like specific thing I remember, I will just mention it. I might be wrong, just don't take my word. So aggression, hyperuricemia, the HGPRD is one of the mnemonic to remember the symptoms for Lesch-Nyhan syndrome. Orange sand, sodium urate crystals in diaper. So I think by diaper they means these patients are young, and as it says purine defective, so it should be more like congenital deficiency. Uh, uh, so gout dystonia, macrocytosis. Treatment is allopurinol or febuxostat is the second line. As here you can see, we are just inhibiting the pathway over here so that there's no uric acid formation. In genetic code features, unambiguous, each codon specifies only one amino acid. So degenerate, reductant, most amino acids are coded by multi-codon. So vowel codon that differ in third vowel position may code for the same tRNA. Amino acid specific base pairing is usually required only in the first two nucleotide position of mRNA codon. Exception is methionine, which is the start codon AUG, and tryptophan UGG, and encoded by only one codon, so it's the same. Comalis, non-overlapping, read from a fixed starting point as a continuous sequence of bases. Exceptions, some viruses. Universal genetic code is conserved throughout evolution. So exception in humans like mitochondria. So universal means it's the same code for all of them. It's not changing for anyone. DNA replication. Eukaryotic DNA replication is more complex than in prokaryotes, but uses many enzymes analogous to those listed below. In both prokaryotes and eukaryotes, DNA replication is semi-conservative, involves continuous and discontinuous Okazaki fragment synthesis, and occurs in the 5' to 3' direction. Origin of replication. So this is a very interesting and very important figure. You will find a lot of animated videos online. I do recommend you go and watch them. Once you watch them, you will never forget it, and you should not forget it. It's very important. Origin of replication. I'll just read here. I will not explain. it much, but I think you should go and watch this video to just refresh your mind if you're done with your first years and all those years. Particular consensus sequence in genome where DNA replication begins may be single, prokaryote, or multiple like eukaryotes. AT-rich, so arginine and thymine-rich sequence such as DATA box region are found in promoter and origin of the replication. Replication fork, which is over here, see replication fork. So, which is a Y-shaped region along DNA temple where leading and lagging strands are synthesized. Helicase, which is the structure C over here, this one, is basically cutting and opening and unwinding. So as it says, unwinding DNA template at replication fork. It's like cutting a cloth, how you are making like two pieces of the cloth by scissor. So helicase is more like scissor. Helicase halves DNA deficiency in Bloom syndrome, BLM gene mutation. So single-stranded binding protein, which is the D over here, see single-stranded binding protein, prevents strands from re-annealing. So you don't want them to coil or tangle. So there's someone who is having their back. So DNA topoisomerase create a single or double strand break in the helix to add or remove supercoils. In eukaryotes, irinotecan or topotecan inhibit topoisomerase top 1 etoposide and tinoposide inhibit top 2. I don't know if I'm pronouncing it right, but sorry. In prokaryotes, fluoroquinolone inhibit top 2 DNA gyrase and top 4. Primase is F, figure F over here, this one, this big guy. Makes an RNA primer on which DNA polymerase 3 can initiate replication. DNA polymerase 3, which is this one, prokaryotes only. These are prokaryotes only. So elongate leading strand by adding deoxynucleotide to 3' and elongates lagging strand until it reaches primer of preceding fragment. DNA polymerase 3 has 3' to 5' to 3' synthesis and proofreading with 3' to 5' exonuclease. Drug blocking DNA replication often have a modified 3' OH, thereby preventing addition of the next nucleotide chain termination. DNA polymerase 1, prokaryote only, degrades the RNA primer, replaces it with DNA. Same functions as DNA polymerase 3 also access RNA primer with 5' to 3' exonuclease. DNA ligase, which is figure I over here, catalyzes the formation of phosphodiester bond within a strand of double-stranded DNA. Join Okazaki fragment ligase link DNA. Telomerase is eukaryote only, a reverse transcription, that is RNA-dependent DNA polymerase that adds DNA, that is TTAGGG to 3' ends of chromosome to avoid loss of genetic material with every duplication. So often deregulated in cancer cells, allowing unlimited replication. So often dysregulated in cancer cells means this is basically like uh, police kind of work. But if this police is not available, then cancer cells have their good time. Telomerase, TAG for greatness and glory. This is one of mnemonic they came up with. Here you have mutation in DNA, severity of damage, that is silent, then missense, nonsense, and frame shift. These are the type of uh, mutations in DNA. There are four types. Types of single nucleotide point mutation is transition purine to purine, that is A to G, or pyrimidine to pyrimidine. Transversion purine to pyrimidine, that is A to D, or pyrimidine to purine, that is C to G. So this is like examples. You can have transition, that is same type, that is purine, or pyrimidine, or transversion, as you can make out from the word, it's like your version is different. The purine to pyrimidine. Single nucleotide substitutions. Silent mutation is nucleotide substitution code for same synonymous amino acid, often base change in third position of codon, tRNA wobble. Missense mutation is nucleotide substitution result in changed amino acid called conservative if new amino acid has similar chemical structure. Example including sickle cell disease, substitution of glutamic acid with valine. Nonsense mutation is really nonsense because nucleotide substitution results in early stop codon. So, or like you're making it. Oh, how did I stop? And there's like, of course, it's a nonsense mutation. So these are the amino acid codons, which are the stop codons, usually result in non-functional proteins. Stop the nonsense. So other mutation is frame shift mutation, which is a deletion or insertion of number of nucleotides not divisible by 3, misreading of all nucleotides downstream. Protein may be shorter or longer, and its function may be disrupted or altered. Example include Duchenne muscular dystrophy, Tay disease, um, uh, splice site mutation, retain intron in mRNA protein with impaired or altered function. Examples include rare causes of cancer, dementia, epilepsy, some type of beta thalassemia, Gaucher's disease, and Marfan syndrome. Here are the silent mutation, some of the examples in picture animation.